Okay guys, good morning everyone. Today we are discussing about bacterial reproduction. Okay, today class bacteria Commonly bacteria show three methods of reproduction. There are first one is binary vegetation reproduction. Next one is asexual reproduction. Third one is sexual reproduction. Vegetation reproduction. Let us see one by one. So vegetation reproduction includes budding and binary fission is the vegetation reproduction, and binary fission takes place during favorable conditions. See on cell division called binary fission. We can see picture binary fission. Okay guys. The bacterial cell divides by a constriction into two halves. Nuclear material also divides into two equal halves. Okay guys, this is the bacterial cell. During binary fission, bacteria is divided into two equal cells. Genetic material also present. Okay. This is divided into two halves bacterial cell. The next one is asexual reproduction. Asexual reproduction takes place by endospores formation conidae and zoospores. These are two ways of sexual reproduction. During unfavorable condition, highly resistant single spores is formed inside the bacteria cell, which is known as endospores. Endo means inside or with spores. Endospores is having a characteristic structure having outer thin exosporium followed by one or many layered spores coated followed by cell wall membrane and matrix. Maybe because of having these much layers, it was highly resistant to endospores is highly resistant to very high and very low temperature, strong chemical and acids etc. due to calcium, dipicholinic acid and peptidoglycan in cortex dipicholinic acid DPA helps into stabilizing its proteins. The next one is sexual reproduction. Occur in the form of genetic recombination. Sexual reproduction nothing but genetic recombination. There are three main methods of genetic recombination: transformation, transduction and conjugation. There are the main methods of genetic recombination. Transformation. Let us see the reproduction. Here transformation. Here genetic material of one bacterial cell goes into another bacterial cell by some unknown mechanism and convert one type of bacterium into another type of bacterium. Non-capsulated bacteria convert into capsulated bacteria and form of capsulated bacteria. Okay guys, see here we can see the genetic material of one bacteria to origin go. ఇది recipient cell formation of DNA. Origin goes to into. This was first studied by Griffith 1928. This was first studied by Griffith. Frederick Gift 1928. Streptococcus pneumonia. Streptococcus pneumonia is known as Griffith effect transduction. In this method, genetic material of one bacterial cell goes to another bacterial cell by agency of bacteriophages or viruses infecting bacteria. Transduction was first of all reported in Salmonella typhimurium by Ginder and Lederberg 1951. In abortive transduction, the gene does not integrate with the recipient genome is lost. Conjugation. Next one is conjugation. We can see bacterial cells in conjugation. Okay. Conjugation. Cell to cell union occur between two bacterial cells, and genetic material DNA of one bacterial cell goes to another cell lengthwise is through conjugation tube, which is formed by sex pili. Conjugation was first reported by Leder 1946. Lederberg. Conjugation occur between donor cell and recipient cell. Conjugation occur between donor cell and recipient cell. This is donor cell, recipient cell. This is donor cell, recipient cell. Okay, the next one is some bacteria that cause human diseases are Clostridium tetanus, Clostridium botulism, Vibrio cholera, Salmonella typhoid, Corynebacterium diphtheria, Mycobacterium tuberculosis, Diplococcus pneumoniae, Mycobacterium leprosy, Neisseria gonorrhea, Treponema pallidum, syphilis. Bacteria also cause certain plants disease like blight of rice, Xanthomonas oryza, citrus conker, corona gall. Okay, bye. Today class